Guys, welcome to the new video. Today I'm gonna cover chapter 1 of microbiology. Now, I know I already made a video explaining chapter 1, but this video is a more detailed, basically more better video than what I did on the first video of chapter 1. And so yeah, let's just go over it. So, the early years of microbiology. What does life really look like? Was a question asked by Anthony van Leeuwenhoek. Um, Anthony van Leeuwenhoek was born in Delft, Netherlands, and he was driven by the curiosity and a desire for self-reliance. And as a cloth merchant, he learned to make his own lenses, which led him to closely examine everyday objects like insect, blood, and his skin. His dedication to observing and recording details made him a pioneer in microscopy and contribution to his lasting fame. So, in other words, he began making and using simple microscopes. He often made a new microscope for each specimen, and he examined water and visualized tiny animals, fungi, algae, and single-celled protozoa called nanomolecules. And by the end of the 19th century, these organisms were called the microorganism. Now, microorganism is an organism that are too small to be seen without a microscope. So here's an image of Anthony van Leeuwenhoek. Uh, he was the first to report the existence of protozoa in 1674 and the bacteria in 1676. So then the question becomes, why did Van Leeuwenhoek discover protozoa before bacteria? And that's because protozoas are eukaryotes, which are generally larger than bacteria, which are prokaryotes. So in terms of size, eukaryotes are bigger than prokaryotes. So Van Leeuwenhoek's simple device, resembling a magnifying glass with screws for adjusting the specimen, revolutionized our understanding of the world. The tiny convex lens, about the size of a pinhead, allowed him to view objects mounted on a specimen holder or in a small glass tube. This tool enabled groundbreaking discoveries in microscopy. So the microbial world. Van Leeuwenhoek reported seeing a scene very much like this picture. So how can microbes be classified? Microbes or microorganisms are the same thing, which means they can't be seen without a microscope. So how can they be classified? Well, Carolus Linnaeus developed a taxonomic system for naming plants and animals and grouping similar organisms together. So Leeuwenhoek's microorganisms can be grouped into six categories, which are bacteria, archaea, fungi, protozoa, algae, and small multicellular cellular animals. So with bacteria and archaea, they're also known as prokaryotes. Fungi, protozoa, algae, and small multicellular cellular animals are known as eukaryotes. So bacteria and archaea. Bacteria and archaea are prokaryotes, and prokaryotes are any unicellular microorganism that lack a nucleus. They are much smaller than eukaryotes, and they are found everywhere where there is sufficient moisture, and some even in isolated extreme environments. They reproduce asexually, meaning that the organism can reproduce on its own. The bacterial cell wall contains peptidoglycan, though some lack cell walls. So when it is said that bacterial cell walls contain peptidoglycan, it means that most bacteria have a rigid outer structure made of a substance called peptidoglycan, which provides strength and shape to the cell. Now, when it says that though some lack cell walls, it means that some bacteria lack cell walls altogether. And these bacteria do not have peptidoglycan structures and often have different strategies for maintaining their shape and integrity. And archaeal cell walls are composed of polymers other than peptidoglycan. So here's an image of cells of the bacterium Streptococcus, which is in dark blue, and two human cheek cells. So notice the size difference between the prokaryotes and eukaryotes. So the prokaryotic bacterial cells right here, and the nucleus, just the nucleus of a eukaryotic cheek cell. So fungi. Fungi is a eukaryote. Eukaryotes are any microorganisms made up of cells containing a nucleus composed of genetic material surrounded by a distinct membrane. And these include animals, animals, plants, algae, fungi, and protozoa once again. They often obtain food from other microorganisms and they possess cell walls. And fungi include these two subgroups, which are mold and yeast. Molds are multicellular, meaning that they are made up of more than one cell. They grow as long filaments and they reproduce by sexual and asexual spores. We then move on to yeast. Yeast are unicellular, meaning that they are made up of one cell only. They reproduce asexually by budding. Budding is a process in which a daughter cell grows off the mother cell. And some produce sexual spores. So the image here shows a fungi. Now image A shows a mold, which produces penicillin that have long filaments that intertwine to form its body, though in this picture it's not shown here, and they reproduce by spores. Image B shows a yeast. Yeast are round to oval and typically reproduce by budding. So protozoa. Protozoa are single-celled eukaryotes. They are similar to animals in nutrient needs and cellular structure. Some live freely in water, and some live in animal hosts. They mostly reproduce asexually, but some also reproduce sexually. Most are capable of locomotion, which means that they have the ability to move from one place to another. And most are capable of locomotion by pseudopods, which are cell extensions that flow in directions of travel, cilia, which are numerous short protrusions that propel organisms through its environment, and flagella, which are extensions of a cell that are fewer, longer, and more whip-like than cilia. So here's an image of a pseudopod, here's an image of a cilia, and here's an image of a flagella. We then also have algae. Algae can be unicellular or multicellular. They are photosynthetic, meaning they make their food from CO2 and H2O using sunlight energy. And they have simple reproductive structures. And algae are categorized on the basis of pigmentation and composition of their cell wall. So image A shows a spirogyra. This microscopic alga grow as change of cells containing helical photosynthetic structures. Image B shows a diatom. These beautiful algae have glass-like cell walls. So there are other microorganisms of importance to microbiologists. So we first have parasites. Microbiologists also study parasitic worms, which range in size from microscopic form to adult tapeworms over 10 meters. Even though most parasitic worms are not microscopic as adults, many of them cause diseases that were studied by early microbiologists. So microbiology books and classes often discover parasitic worms. Further, laboratory scientists diagnose infections of parasitic worms by finding microscopic eggs and immature stages in blood, fecal, urine, and lymph specimen. The only type of microbe that remained hidden from van Leeuwenhoek and other microbiologists was the virus, which are typically much smaller than the smallest prokaryote and is not usually visible by light microscope. Viruses were not seen until the electron microscope was invented in 1932. All viruses are acellular, which means they are not composed of cells, and obligatory intracellular parasites composed of small amounts of genetic material, either DNA or RNA, surrounded by a protein coat. So this image here shows an immature stage of a parasitic worm in blood. Then this image here shows a colorized electron microscope of an image of a virus infecting a bacterium. So what you should take away from this image is the size difference between a virus compared to a bacterium. So the golden age of microbiology. So the competition among scientists who were striving to be the first to answer these four questions drove the explanation/discovery in microbiology during the late 1800s through the early 1900s. So scientists searched for answers to these four questions. We first have, is spontaneous generation of microbial life possible? So some philosophers and other scientists of the past thought that living things arose from these three processes: asexual reproduction, sexual reproduction, and non-living matter. Now, Aristotle proposed the idea of spontaneous generation, which also means non-living matter right here, in which living things can arise from non-living things. So Reddy's experiment. Now, I'm going to come back to this slide because I think it's better to show you in pictures. So Reddy's experiment was designed to challenge the theory of spontaneous generation. Specifically, Reddy tested the belief that maggots spontaneously emerge from rotting meat. So here's the setup. We have three flasks. One flask is open. Flask 2 is sealed. And flask 3 is covered with glaze. So when the flask remained unsealed, maggots covered the meat within a few days. When the flask was sealed, flies were kept away and no maggots appeared on the meat. And when the flask opening was covered with glaze, flies were kept away from the meat and no maggots appeared there, though a few appeared on top of the glaze. So, in other words, when decaying meat was kept isolated from flies, maggots never developed. And when the meat was exposed to flies, the meat was soon infested. And as a result, scientists began to doubt Aristotle's view. So we then have Needham's experiment. Scientists agreed that large animals could not arise spontaneously, but believed that microbes could. Needham experiment with beef gravy and infusion of plant materials reinforced this idea. So, in the 18th century, Needham conducted an experiment to support the theory of spontaneous generation. He boiled beef broth and plant fusions to kill any living microorganisms and then sealed the vials with corks. However, after a few days, he noticed that the vials became cloudy and microscopic organisms were present. Based on this, Needham concluded that life must spontaneously arise from non-living matter, as he believed that heat had killed all pre-existing life in the vials. Needham findings seem to support the existence of life force that could cause inanimate matter to come to life, which impressed many in the scientific community at the time, including the prestigious Royal Society in Britain, which made him a member. However, his experiments were later challenged by scientists like Lazzaro Spallanzani, who suggested that Needham methods might not have been rigorous enough to eliminate all forms of life before sealing the vials. So we have Spallanzani's experiment. His experiment contradicted with Needham findings, and he concluded that Needham failed to heat vials sufficiently to kill all microbes or had not sealed them tightly enough. Microorganisms exist in air and can contaminate experiment. Spontaneous generation of microorganisms does not occur. All living things arise from other living things. However, critics said that sealed vials did not allow enough air for organisms to survive and that prolonged heating destroyed the life force. So when critics say that sealed vials did not allow enough air for organisms to survive, it is because at the time, many people believed that air contained an essential element, sometimes referred to as the life force, is necessary for life to spontaneously generate. The critics argued that sealing the vials so tightly prevented the air, thus the supposed life force, from entering, which they believed was needed for organisms to form and survive. They suggested that the lack of air, rather than the absence of spontaneous generation, was the reason for no microbial growth in Spallanzani's experiment. And when critics said that the prolonged heating destroyed the life force, it meant that some critics also believed that Spallanzani's extended boiling of the infusion might have destroyed the mystical life force present in the substance, preventing spontaneous generation. So, in other words, they suggested that the heat might have eliminated not just existing life, but also whatever the unseen force of condition might be necessary for new life to arise from non-living matter. So then we have Pasteur's experiment. Now, I'm going to come back to this slide again because I'm going to show you in picture. So Pasteur's experiment with swan neck flask. Pasteur boiled the broth or infusion inside a glass flask to kill any pre-existing microorganism, ensuring the broth was completely sterile. The flask neck was bent into an S shape, in which allowed air to enter the flask, satisfying the belief that air was necessary for life to form. However, the S shape trapped the dust and the airborne particles in its curve, preventing them from reaching the broth. After the infusion cooled, air could move freely in and out of the flask, but no microbes appeared in the broth, even after extended periods. So as long as the neck of the flask remained intact, the broth stayed sterile, showing that the introduction of microorganisms from the air was necessary for microbial growth. The curve in the neck flask trapped the dust and microbes, preventing contamination of the broth. This demonstrated that microorganisms from the environment were responsible for microbial growth and not spontaneous generation. So, in other words, when the flask remained upright, no microbial growth appeared. But when the flask was tilted, dust from the bend in the neck seeped into the back of the flask and made the infusion cloudy with microbes within a day. So, the scientific method. So, the debate over spontaneous generation led in part to the development of scientific method, in which observation leads to question generates hypothesis, and the hypothesis is tested through experiments, and the results prove or disprove a hypothesis. The accepted hypothesis leads to theory/law, and disproved hypothesis is rejected or modified. So this is the framework for scientific research. So one is a group of observation that leads a scientist to ask a question about a phenomenon. Two is when the scientist generates a hypothesis, that is a potential answer to the question. Three, the scientist designs and conducts an experiment to test the hypothesis. And four is based on the observed results from the experiment, the scientists either accept, reject, or modify the hypothesis. So then we move on to the second question. What causes fermentation? So, spoiled wine threatened the livelihood of many grape growers. Wine producers funded research of methods to promote the production of alcohol and prevent spoilage during fermentation. The debate over the cause of fermentation reaction was also linked to the debate over spontaneous generation. So the debate over the cause of fermentation was linked to the debate over spontaneous generation because both centered on whether life and biological process could arise from non-living matter. Some scientists believe that fermentation was purely a chemical process or caused by air, much like the idea that microorganisms could spontaneously appear. Louis Pasteur's experiment proved that fermentation was caused by the living organisms like yeast and bacteria, and that these organisms did not spontaneously generate but came from pre-existing life. His finding helped disprove both spontaneous generation and the non-biological cause of fermentation. So, Pasteur's experiment. Some scientists believe air caused fermentation. Others insisted that living organisms cause fermentation. So, Pasteur conducted a series of experiments that addressed the cause of fermentation. So, how Pasteur applied the scientific method in investigating the nature of fermentation. So, we look at the hypothesis. Hypothesis 1 states that fermentation occurs on its own without the need for living microorganisms. So, Pasteur heated the grape juice to kill all microbes and sealed the flask. No fermentation occurred and no microbes were observed, rejecting the idea that fermentation occurs spontaneously. Hypothesis 2 states that the presence of air causes fermentation. So the flask was left open via a curved neck to allow the airflow while preventing contaminants. Again, no fermentation occurred, rejecting the idea that air causes fermentation. Hypothesis 3 states that bacteria are responsible for fermenting the juice. So Pasteur introduced the bacteria in the flask. Bacteria were produced, but instead of alcohol, acids were produced. This led to modify the hypothesis showing that bacteria ferment grape juice into acids and not alcohol. And then finally, hypothesis 4. Hypothesis 4 states that yeast is responsible for fermentation. So yeast was introduced into the flask and yeast cell reproduced. Fermentation occurred and alcohol was produced, confirming that yeast is responsible for fermenting grape juice into alcohol. So Pasteur's experiment led to the development of pasteurization, in which the process of heating liquid just enough to kill most bacteria. And this essentially began the field of industrial microbiology, which is the intentional use of microbes for manufacturing products. So some industrial uses of microbes. So if I remember correctly, she said this is just an FYI and not necessarily something that you should remember for the upcoming exam. So Buchner's experiment. Buchner's experiment demonstrated the fermentation does not require living cells, and instead showed enzymes promote chemical reactions. Buchner's work began the field of biochemistry. So, in other words, Buchner's experiment in 1897 demonstrated that fermentation could occur without leaving yeast cells. By using a cell-free yeast extract and mixing it with sugar, Buchner observed that fermentation still produced alcohol, proving that enzymes within the extract and not the living cells were responsible for the process. This discovery debunked the idea that fermentation required living cells and led to the development of biochemistry and the study of metabolism, showing that enzymes drive biochemical reactions independently. And then we move on to the third question. What causes disease? So Pasteur developed the germ theory of disease. The germ theory states that microorganisms are the cause of infectious disease. Some diseases are caused by specific germs called pathogens. Pathogens are microorganisms that are capable of causing disease. So early investigators began to suspect that diseases like cholera, tuberculosis, and anthrax are caused by specific germs. So here's an image of Robert Koch. Koch was an instrumentalist in modifying the scientific method to prove that a given pathogen causes a specific disease. So Koch's experiment. Robert Koch studied the causative agents of disease, which is called etiology, and he demonstrated that a bacterium causes anthrax, and he also examined colonies of microorganisms. So this list are the significant contribution to microbiology and disease causation that was presented by Koch's experiment. So we first have simple staining technique. So these techniques allowed scientists to better visualize bacteria under a microscope. We also have first photomicrograph of a bacteria. So Koch was the first to capture a microscopic image of a bacteria, which was a major step in documenting microorganisms. We also have first photograph of a bacteria in a diseased tissue. So Koch also provided the first image of bacteria present in infected tissues, directly linking bacteria to disease. We then also have techniques for estimating bacterial numbers in a solution. So these methods enabled more accurate studies of bacterial growth and concentration. There's also a use of steam to sterilize growth media. So sterilization by steam, a precursor to modern autoclaving, helped prevent contamination during bacterial culture experiment. And there's also use of Petri dishes. So Koch's lab was instrumental in the development and the use of Petri dishes, which provided a sterile environment to cultivating bacteria. Then there's also laboratory techniques to transfer bacteria. So these are the methods for safely transferring bacteria between mediums were essential for isolating and studying specific bacterium strains. And then finally, we have bacteria as a distinct species. So Koch demonstrated that that different bacteria, are different species. and each are responsible for specific diseases, which laid the groundwork for bacterial classification. So this image here shows Fanny Hesse. Hesse was an instrumentalist in developing the use of agar as a solidifying agent in microbiological media. So this image here shows a bacterial colonies on a solid surface, which is the agar, invented by Fanny Hesse. So differences in colony shape, size, and color indicate the presence of different species. Such differences allowed Koch to isolate specific types of bacteria that could be tested for their ability to cause diseases. So Koch's postulates. So Koch's postulates are a set of criteria established by Robert Koch to scientifically prove that a particular microorganism is the cause of a specific infectious disease. These postulates provide a systemic approach to link pathogens to diseases. So Koch initially developed and applied these steps in his research to anthrax and tuberculosis. So his work on tuberculosis, where he identified Mycobacterium tuberculosis as the causative agent, earned him the Nobel Peace Prize in 1905. So the four postulates are as follows. 1. The suspected causative agent must be found in every case of the disease and be absent from healthy hosts. So the pathogen should be consistently present in the individual suffering from the disease but absent in healthy individuals. 2. The agent must be isolated and grown outside the host. So the microorganisms must be cultured and grown in a laboratory setting, separate from the host to study it further. 3. When the agent is introduced to a healthy, susceptible host, the host should get the disease. So when the isolated pathogen is introduced into a healthy organism, it should cause the same disease, confirming its role in disease causation. And 4, the same agent must be found in the disease experimental host. So after the disease develops in the new host, the same microorganism should be isolated again, proving that it is indeed the cause of the disease. So here are the other notable scientists of the golden age of microbiology and the agents of disease that they discovered. So if I'm not mistaken, she said that we don't have to memorize all of these. I think maybe just a few mentioned in the slides, but not the ones in the entire chart. So gram stain. Gram stain is a technique for staining microbial samples by applying a series of dyes that leave some microbes purple and others pink. And it was developed by Christian Gram in 1884. So the gram stain is the most widely used staining technique and one of the first steps to identify a bacterium. Okay, so here are the results of gram staining. So gram-positive cells, in this case Staphylococcus aureus, are purple, whereas gram-negative cells, in this case E. coli, are pink. We then finally move on to the last question. How can we prevent infection and diseases? So we first have Samuel Wace and hand washing. So Ignaz Semmelweis was a physician in 1848. He discovered that hand washing significantly reduced the incidence of puerperal fever in women giving birth. He hypothesized that medical students were transmitting infectious cadaver particles from autopsies to the delivery rooms. By requiring students to wash their hands with chlorinated lime water, mortality rate dropped from 18.3% to 1.3%. Despite this success, his ideas were rejected by his colleagues, and he was eventually forced to leave his position. Samuel Wace later died in a mental institution from an infection caused by Streptococcus, a bacterium responsible for the fever that he sought to prevent. His work paved the way for the acceptance of antiseptic practices and germ theory. And so then we have Lister's antiseptic technique. So Joseph Lister, an English surgeon, advanced the idea of antisepsis in healthcare by using carbolic acid to disinfect wounds, surgical incisions, and dressings, which significantly reduced infections. Despite the initial resistance, his method reduced patient death by two-thirds, leading to the widespread adoption of antiseptic practices. Lister is regarded as the founder of antiseptic surgery, and his work vindicated Ignaz Semmelweis, paving the way for further research in antisepsis and disinfection in medicine. And so then we move on to Nightingale and nursing. So Florence Nightingale was an English nurse who introduced cleanliness and antiseptic techniques during the Crimean War, which significantly improved the hygiene and saving many lives. She emphasized that cleaning wards and ensuring patient clothes and dressings were sanitized. As a skilled mathematician, she used statistical data to demonstrate that poor hospital conditions were causing many deaths. Her work transformed nursing practices and contributed to the field of epidemiology. So here's an image of Florence Nightingale. She is often referred to as the founder of modern nursing. She was influential in introducing antiseptic technique into nursing practices. And then we move on to the infection control and epidemiology, presented by Snow. So, John Snow was an English physician, and he played a crucial role in establishing public health standards by preventing the spread of infectious disease. In 1854, during the cholera epidemic in London, he traced the outbreak to a contaminated water supply on a broad street. Although he did not know the exact cause, His finding underscored the need for clean water and proper sewage treatment. Snow's work laid the foundation for infection control and epidemiology, the study of disease spread and the distribution. His approach remains fundamental in modern public health. We then move on to the field of immunology, which is Jenner's vaccine. So, Lady Mary Wortley Montagu introduced the practice of inoculation against smallpox to Europe. During her travels in the Ottoman Empire, she learned that inoculating children with pus from a mild case of smallpox could provide protection against the disease. In 1721, upon returning to England, she had her daughter inoculated, marking the first use of the procedure in the country. This practice, known as variolation, was promoted by Lady Montagu as it significantly reduced the risk of death from smallpox in those who underwent it. And so then we move on to the field of chemotherapy, which was formed by Ehrlich's magic bullets. So Paul Ehrlich was a German microbiologist, and he built on the idea that chemicals could be used to target and kill specific microorganisms without harming humans. This led him on a quest to find a magic bullet, a chemical that would destroy pathogens while being harmless to people. In 1908, Ehrlich discovered an arsenic-based drug which is effective against causative agents of syphilis. Though it had serious side effects, his work laid the foundation of the field of chemotherapy, the use of chemicals to treat diseases. So here's a picture of Lady Mary Wortley Montagu. So again, Montagu introduced England to variolization, which is the procedure in which she oculates patients with pus from a mild smallpox case to protect them against a more severe case. So in pre-1857, or before the golden age of microbiology, we first have Van Leeuwenhoek. Van Leeuwenhoek's modern discipline was bacteriology, protozoology, mycology, parasitology, and phycology. And his contributions was that he first observed and described microorganisms, laying the groundwork for these fields. We then also have Linnaeus. Linnaeus' modern discipline was taxonomy, in which he contributed by developing the system of naming and classifying organisms. Then we have Semmelweis and Snow. Um, their modern discipline was infection control and epidemiology. And Semmelweis promoted hand washing to prevent infection, whereas Snow is known for his work in controlling cholera outbreak and establishing the foundation of epidemiology. We then get into the golden age of microbiology, starting off with Pasteur. So Pasteur's modern discipline were in the industrial microbiology and food/beverage technology. And his contribution was that he developed pasteurization and made crucial contribution to microbial metabolism and fermentation. Then we have Buchner. His modern discipline was microbial metabolism, genetics, and genetic engineering. So his contribution was that he demonstrated that fermentation is driven by enzymes, leading to advances in biochemistry and genetics. We also have Koch. His modern discipline was etiology. So his contribution was that he established Koch's postulates, a foundational framework for identifying disease-causing microorganisms. And then we have Graham. Her distribution was in the microbial morphology, and she contributed in the development of the Graham staining technique, which is crucial for identifying and classifying bacteria. Then we have Lister. Um, Lister's modern discipline was antiseptic medical techniques, which is the hospital microbiology, and his distribution was introducing antiseptic practices in surgery, reducing infections. Then we have Nightingale. Her modern discipline was nursing, and her contribution was that she pioneered modern nursing and importance of sanitation in healthcare. And then we move on to Jenner. Now, her modern discipline was immunology, so her contribution was that she developed the smallpox vaccine. And then we move on to Ehrlich. Now, his modern discipline was chemotherapy, and his contribution was that he developed treatments for diseases using chemical agents. And then here is the fields of microbiology. So this is just a FYI, and again, something that she said that you don't have to remember for the upcoming exam. Well, with that being said, that's the end of the video.